Continuation of the cancer lecture. So, tumor suppressor genes and oncogenes, brakes and accelerators. The genes that have been implicated in carcinogenesis are divided into two broad categories: tumor suppressor genes and oncogenes. Tumor suppressor genes act as cell's brakes. They encode proteins that restrain cell growth and prevent cells from becoming malignant. The existence of such genes originally came to light from studies in the late 1960s in which normal and malignant rodent cells were fused to one another. Some of the cell hybrids formed from this type of fusion lost their malignant characteristics, suggesting that a normal cell possesses factors that can suppress the uncontrolled growth of a cancer cell. So, normal cell possesses factors that can suppress the uncontrolled growth of a cancer cell. Further evidence for the existence of tumor suppressor genes are gathered from observations that specific regions of particular chromosomes are constantly deleted in cells of certain types of cancer. If the absence of such genes is correlated with the development of a tumor, then it follows that the presence of these genes normally suppress the formation of tumor. Oncogenes, on the other hand, encode proteins that promote the loss of growth control and the conversion of a cell to a malignant state. Most oncogenes act as accelerators of cell proliferation, but they have other roles as well. Oncogenes may lead to genetic instability. Most oncogenes act as accelerators of cell proliferation, but they have other roles as well. Oncogenes may lead to genetic instability, prevent a cell from becoming a victim of apoptosis, or promote metastasis. The existence of oncogenes was discovered through a series of investigations on RNA tumor viruses that is documented in the experimental pathways at the end of this chapter. This virus transform a normal cell into a malignant cell because they carry an oncogene that encode a protein that interferes with the cell's normal activities. The turning point in this studies came in 1976 when it was discovered that an oncogene called SRC, carried by an RNA tumor virus called an avian sarcoma virus, was actually present in the genome of uninfected cells. So, oncogene example is SRC. SRC is a oncogene. Oncogene in the sense promoting type gene. The turning point in the studies came in 1976. So the oncogene in fact was not a viral gene, but a cellular gene that had become incorporated into the viral genome during a previous infection. It soon became evident that cells possess a variety of genes, now referred to as proto-oncogenes, that have the potential to subvert the cell's own activities and push the cell towards the malignant state. As discussed below, proto-oncogenes encode proteins that have various functions in a cell's normal activities. Proto-oncogenes can be converted into oncogenes activated by several mechanisms. The gene can be mutated in a way that alters the properties of gene product so that it has no longer functions normally. A gene can become duplicated one or more times, resulting in gene amplification and excess production of the encoded protein. And we try to learn about the contrasting effects of mutations in tumor suppressor genes and oncogenes. Whereas a mutation in one of the two copies of an oncogene may be sufficient to cause a cell to lose growth control, both copies of a tumor suppressor gene must be knocked out to induce the same effect. As discussed shortly, oncogenes arise from proto-oncogenes as the result of gain of function mutations. That is, mutations can cause the gene product to exhibit new functions that lead to malignancy. Tumor suppressor genes in fact suffer loss of function mutations and or epigenetic inactivation that render them unable to restrain cell growth. So, contrasting effects of mutation. Mutation in one of the two copies of an oncogene may be sufficient to cause a cell to lose growth control, but copies of a tumor suppressor gene must be knocked out to induce the same effect. As discussed shortly, oncogenes arise from proto-oncogenes as a result of gain of function mutations, that is, mutations that cause the gene product to exhibit new functions that lead to malignancy. Tumor suppressor genes, in contrast, suffer loss of function mutations or epigenetic inactivation that render them unable to restrain cell growth. So, mutated tumor suppressor gene, normal cell growth, and loss of growth control. Copies of mutated tumor suppressor gene on both homologues. Then, normal cell growth. Proto-oncogene, normal cell growth will be happening. Then, mutated proto-oncogenes has become oncogenes. Then, loss of growth control will be happening. Then, activation of a proto-oncogene to an oncogene. Activation can be accomplished in several ways, and in pathway A, a mutation in the gene alters the structure and function of encoded protein. In pathway B, gene amplification results in overexpression of the gene. In pathway C, arrangement of DNA brings a new DNA segment into the vicinity or up against the gene, altering either its expression or the structure of the encoded protein. So, regulatory region, proto-oncogene, protein encoded by proto-oncogene. There are A, B, C, three kind of pathways. In case of A, mutation or deletion is happening, so encoded protein with altered structure or function will be formed. But in case of B, gene duplication, increased synthesis of encoded protein will be happening. Then in C, a DNA regulatory sequence translocated from distant sites alters expression of downstream gene. Then increased synthesis of encoded proteins can be seen. Then synthesis of a protein containing portions encoded by different genes. The fusion protein is no longer under normal control. A protein coding gene translocated from distant sites fuses with portion of gene, causing formation of a fusion gene. Then third method, a chromosome rearrangement can occur that brings a DNA sequence from a distant site in the genome into close proximity of the gene, which can either alter the expression of the gene or the nature of the gene product. Any of these genetic alterations can cause a cell to become less responsive to normal growth controls. Oncogenes act dominantly, which is to say that a single copy of an oncogene can cause the cell to express the altered phenotype regardless of whether or not there is a normal inactivated copy of the gene on the homologous chromosome. Researchers have taken advantage of this property to identify oncogenes by introducing the DNA suspect of containing the gene into cultured cells and monitoring the cells for evidence for altered growth properties. We saw earlier that the development of a human malignancy requires more than a single genetic alteration. The reason becomes more apparent with the understanding that there are two types of genes responsible for tumor formation. As long as a cell has its full complement of tumor suppressor gene, it is thought to be protected against the effects of an oncogene for reasons that will be evident when the functions of these genes are discussed below. Most tumors contain alterations in both tumor suppressor gene and oncogene, suggesting that the loss of tumor suppressor function within a cell must be accompanied by the conversion of a proto-oncogene into an oncogene before the cell can become fully malignant. Even then, the cell may not exhibit all of the properties required to invade surrounding tissues or to form secondary colonies by metastasis. Then, mutations in additional genes such as those encoding cell adhesion molecules or extracellular proteases may be required before the cells acquire a full life-threatening phenotype. We can now turn to the functions of the products X encoded by both tumor suppressor genes and oncogenes and examine how mutations in these genes can cause a cell to become malignant. Then, tumor suppressor genes. The transformation of a normal cell to a cancer cell is accompanied by loss of function of one or more of tumor suppressor genes. At the present time, more than 2 dozen genes have been implicated as tumor suppressors in human, some of which are listed in table 16.1. So, which are they? APC, BRCA1, MSH2, MLH2, E-cadherin, INK4A, NF1, NF2, TP53, P10, RB, VHL, WT1. So, APC is a primary tumor of colorectal cancer. Proposed function binds beta-catenin acting as transcription factor that causes inherited syndrome called familial adenomatous polyposis. And then BRCA1, breast cancer, DNA repair, familial breast cancer. Then MSH2 and MLH2, colorectal cancer. Then mismatch repair will be the proposed function. Then HNPCC is an inherited syndrome related to this. Then E-cadherin, colon cancer, breast cancer, cell adhesion molecule is the proposed function, but that may lead to inherited syndrome called familial gastric cancer. Then melanoma, pancreatic cancer, pancreatic primary tumor will be produced because of INK4A. And the proposed function of that INK4A gene is P16 CDK inhibitor. ARF stabilizes P53, but that might end up having inherited syndrome called familial melanoma. Then NF1 primary tumor is neurofibromas. Then proposed function of NF1 is activates GTPase of RAS. That will lead to inherited syndrome called neurofibromatosis type 1. Then NF2 gene, meningiomas. So proposed function was links membrane to cytoskeleton, but that will end up having neurofibromatosis type 2. That's an inherited syndrome. Then P10, breast thyroid cancer, PIP3 phosphate is the proposed function. Then Cowden disease is an inherited syndrome related to that. Then RB, retinal, binds E2F, cell cycle transcription regulation, retinoblastoma. Then VHL, kidney protein ubiquitination and degradation, von Hippel-Lindau syndrome. Then WT1, Wilms tumor of kidney, transcription factor, Wilms tumor. Included among the genes on the list are those that encode transcription factors for TP53 and WT1, cell cycle regulators like RB and INK4A, components that regulate G proteins, NF1 and phosphoinositide phosphatase, P10, and a protein that regulates protein degradation, VHL. In one way or another, most of the proteins encoded by tumor suppressor genes act as negative regulators of cell proliferation, which is why their elimination promotes uncontrolled cell growth. The products of tumor suppressor genes also help maintain genetic stability, which may be a primary reason that tumors contain such an aberrant karyotype. Some tumor suppressor genes are involved in the development of a wide variety of different cancers, whereas others play a role in the formation of one or a few cancer types. It is common knowledge that members of some families are at high risk of developing certain types of cancers. Although the inherited cancer syndromes are rare, they provide an unprecedented opportunity to identify tumor suppressor genes that, when missing, contribute to the development of both inherited and sporadic or non-inherited forms of cancer. The first tumor suppressor gene to be studied and eventually cloned, and one of the most important, is associated with the rare childhood cancer of the retina of the eye called retinoblastoma. The gene responsible for this disorder is called RB. The incidence of retinoblastoma follows two distinct patterns. It occurs at high frequency and at young age in members of certain families, and it occurs sporadically at an older age among members of the population at large. The fact that retinoblastoma runs in certain families suggested that the cancer can be inherited. Examination of cells from children suffering from retinoblastoma revealed that one member of a 13th pair of homologous chromosomes are missing, a small piece from the interior portion of the chromosome. The deletion was present in all of the children's cells, both the cells of the retinal cancer cells and cells elsewhere in the body, indicating that the chromosomal aberrations had been inherited from one of the parents. Retinoblastomas is inherited as dominant genetic trait because members of high risk families that develop the disease inherit one normal allele and one abnormal allele. But unlike most dominantly inherited conditions such as Huntington's disease, where an individual who inherits a missing or an altered gene invariably develops the disease, children who inherit a chromosome missing the retinoblastoma gene inherit a strong disposition toward developing retinoblastoma rather than the disorder itself. In fact, approximately 10% of individuals who inherit a chromosome with an RB deletion never develop the retinal cancer. How is it that a small percentage of these predisposed individuals escape the disease? The genetic basis of retinoblastoma was explained in 1971 by Alfred Knudsen of the University of Texas. Knudsen proposed that the development of retinoblastoma requires that both the copies of the RB gene of a retinal cell are either eliminated or mutated before the cell can give rise to an retinoblastoma. In other words, the cancer arises as a result of two independent hits in a single cell. In cases of sporadic retinoblastoma, the tumor develops from a retinal cell in which both copies of RB gene have undergone successive spontaneous mutation. Because the chance that both alleles of the same gene will be the target of debilitating mutations in the same cell is extremely unlikely, the incidence of the cancer in the general population is extremely low. In contrast, the cells of a person who inherits a chromosome with an RB deletion are already halfway along the path to become malignant. Mutations or deletions of the remaining RB allele in any of the cells of the retina produces a cell that lacks a normal RB gene and thus cannot produce a functional RB gene product. This explains why individuals who inherit an abnormal RB gene are so highly predisposed to developing the cancer. The second hit fails to occur in approximately 10% of these individuals who do not develop the disease. Knudsen's hypothesis was subsequently confirmed by examining cells from patients with an inherited disposition to retinoblastoma and finding that, as predicted, both alleles of the gene were missing or mutated in the cancer cells. Individuals with sporadic retinoblastomas had normal cells that lacked RB mutations and tumor cells in which both alleles of the gene were mutated. Although deficiencies in the RB gene are first manifested in the development of retinal cancers, it is not the end of the story. People who suffer from the inherited form of retinoblastoma are also at a high risk of developing other types of tumors later in life, particularly soft tissue sarcomas or tumors of mesenchymal rather than epithelial origin. The consequences of RB mutations are not confined to persons who inherit a mutant allele. Mutations in RB alleles are a common occurrence in sporadic breast, prostate, and lung cancers among individuals who have inherited two normal RB alleles. When cells from the tumors are cultured in vitro, the reintroduction of a wild-type RB gene back into the cells is generally sufficient to suppress their cancerous phenotype, indicating that the loss of this gene function contributes significantly to tumor genesis. Let's look some more closely at the role of the RB gene. Mutations in the RB gene that can lead to retinoblastoma. In sporadic, non-familial cases of the disease, an individual begins life with two normal copies of the RB gene and the zygote, and retinoblastoma occurs only in those. An individual begins life with two normal copies of RB gene in the zygote, and retinoblastoma occurs only in those rare individuals in whom a given retinal cell accumulates independent mutations in both alleles of the gene. In familial or inherited case of the disease, an individual begins life with one abnormal allele of the RB gene, usually present as a deletion. Thus, all the cells of the retina have at least one of their two RB genes non-functional. If the other RB allele in the retinal cell becomes inactivated, usually as the result of a point mutation, that cell gives rise to retinal tumor. So the role of PRB in regulating the cell cycle. The importance of the cell cycle in cell growth and proliferation was very important, where it was noted that factors that control the cell cycle can play a pivotal role in the development of cancers. In its best studied role, the protein encoded by the RB gene, PRB, helps regulate the passage of cell from G1 stage of a cell cycle into S phase, during which DNA synthesis occurs. As the transitions from G1 to S is a time to commitment for the cell. Once the cell enters S phase, it invariably proceeds to the remainder of the cell cycle and into mitosis. The transition from G1 to S is accompanied by the activation of many different genes that encode proteins ranging from DNA polymerase to cyclins and histones. Among the transcription factors involved in activating genes required for S phase activities are members of the E2F family of transcription factors, which are key targets of PRB. A model depicting the role of PRB in controlling is very important. During G1, E2F proteins are normally bound to PRB, which prevents the E2F molecules from activating a number of genes encoding proteins required for S phase activities like cyclin E and DNA polymerase alpha. Studies suggest that the E2F PRB complex is associated with DNA but acts as a gene repressor rather than a gene activator. As the end of G1 approaches, the PRB subunit of the PRB E2F complex is phosphorylated by the cycling-dependent kinase that regulate the G2S transition. Once phosphorylated, PRB releases its bond to E2F, allowing the transcription factor to activate gene expression, which marks the cell's irreversible commitment to enter S phase. A cell that loses PRB activity as a result of RB mutation would be expected to lose its ability to inactivate E2F, thereby removing certain restraints over the entry to S phase. So E2F is only one of dozens of proteins capable of binding to PRB, suggesting that PRB has numerous other functions. The complexity of PRB interactions is also suggested by the fact that the protein contains at least 16 different serine and threonine residues that can be phosphorylated by cycling-dependent kinases. It is likely that phosphorylation of different combinations of amino acid residues allows the protein to interact with different downstream targets. The importance of PRB as a negative regulator of cell cycle is demonstrated by the fact that DNA tumor viruses, including adenovirus, human papillomavirus, and SV14, encode a protein that binds to PRB, blocking its ability to bind to E2F. The ability of these viruses to induce cancer in infected cells depends on their ability to block the negative influence that PRB has on progression to cell through the cell cycle. By using this PRB blocking proteins, these viruses accomplish the same result as when the RB gene is deleted, leading to the development of human tumors. Then the role of PRB in controlling transcription of genes required for progression of cell cycle. During most of G1, the unphosphorylated PRB is bound to E2F protein. The E2F PRB complex binds to regulatory sites in the promoter regions of numerous genes involved in cell cycle progression, acting as a transcriptional repressor that blocks gene expression. Repression probably involves the methylation of lysine 9 of histone H3 that modulates chromatin architecture, and activation of cycling dependent kinase or CDK leads to the phosphorylation of PRB, which can no longer bind the E2F protein. In the pathway depicted here, loss of the bound PRB converts the DNA bound E2F into a transcriptional activator, leading to expression of the gene being regulated. The mRNA is translated into proteins that are required for progression of cells from G1 to S phase of the cell cycle. Other roles of PRB have been identified but are not discussed. Then the role of P53, guardian of the genome. The TP53 gene may have more to do with the development of human cancer than any other component of the genome. The gene gets its name from the product 10 goats P53, which is a polypeptide having the molecular mass of 53,000 Daltons. In 1990, TP53 was recognized as the tumor suppressor gene that, when absent, is responsible for a rare inherited disorder called the Li-Fraumeni syndrome. Victims of this disease are afflicted with a very high incidence of various cancers, including breast and brain cancer and leukemia. Like individuals with the inherited form of retinoblastoma, persons with this syndrome inherit Li-Fraumeni syndrome. Inherit one normal and one abnormal or deleted allele of the P53 tumor suppressor gene, and others highly susceptible to cancers that result from random mutations in the normal allele. The importance of P53 as an anti-tumor weapon is most evident from the fact that TP53 is most commonly mutated gene in human cancers. Approximately half of all human tumors contain cells with point mutations or deletions in both alleles of the TP53 gene. Furthermore, tumors composed of cells bearing TP53 mutations are correlated with a poor survival rate than those containing a wild-type TP53 gene. Clearly, the estimation of TP53 function an important step in the progression of many cancer cells toward the fully malignant state. So the role of tumor suppressor gene TP53 in human cancers, the frequency with which both alleles of the TP53 gene are mutated in different types of cancers. The data differs to the most common form of each of these 10 types of cancers. P53 function is particularly sensitive to mutations in its DNA binding domain. P53 functions as a tetramer, each subunit of which consists of several domains with different functions. This image shows a ribbon drawing of DNA binding domain. 6 amino acid residues, most often mutated in P53 molecules that have been debilitated in human cancers, are indicated in a single letter nomenclature. These residues occur at or near the protein-DNA interface and either directly impact the binding of the protein to DNA or alter its conformation. Why is the presence of P53 so important in preventing a cell from becoming malignant? For one thing, P53 seems to bind a very long list of different proteins as well as DNA and is involved in a diverse array of cellular activities. In its best studied role, P53 serves as a transcription factor that acts as a crucial player in a cell's response to stress. When a cell sustains DNA damage, P53 responds by altering the expression of a large number of genes involved in cell cycle regulation, apoptosis, or senescence. The importance of the transcription regulating role of P53 is very important, which shows the location of the 6 mutations most commonly found to disable P53 in human cancers. All of them map in the region of the protein that interacts with the DNA. One of the best studied genes activated by p53 includes a protein called p21 that inhibits the cycling dependent kinase that normally drives a cell through the G1 checkpoint. As the level of p53 rises in the damaged G1 cell, expressions of the p21 gene is activated and progression through the cell cycle is arrested. This gives the cell time to repair the genetic damage before it initiates DNA replication. When both copies of the TP53 gene in the cell have been mutated so that their product is no longer functional, the cell can no longer produce the p21 inhibitor or exercise the feedback control that prevents it from entering a phase when it is not prepared to do so. Failure to repair DNA damage leads to the production of abnormal cells that have the potential to become malignant. Next topic we will be learning with the help of next video.